Biology 9700, paper 2, variant 2, May June 2024, question number 4 to question number 6. And this is the second video. Question number 4, in the mesophyll tissues of the leaves, products of photosynthesis can be used to synthesize organic compounds such as the polysaccharide, cellulose, and some amino acids. A source of nitrogen for amino acid synthesis can be provided by nitrate ions that have been taken up in the roots and transported to the leaves. Describe the structure of a cellulose molecules. And students, you know, the cellulose is the polysaccharide, uh, which is formed when in the form linear chains. It is in the form of linear chains of glucose. And which glucose? This is the beta glucose, not alpha glucose. This is the beta glucose. And you know, the structure of beta glucose is just like this, in which OH is in the upward direction. So linear chains of glucose or the beta glucose, they will join. So many beta glucose molecules, they will join together to form one C1 and C4. They will join together to make a glycosidic bond between carbon C1 and C1. Formation of glycosidic bond will take place by the condensation reaction. And what is condensation reaction? Condensation reaction is that reaction in which on the formation of a bond, one water molecule is removed. So when there will be the bond, glycosidic bond between C1 and C4, then one water molecule is removed. So it means that on every addition or on the formation of every bond, the one water molecule is removed, and this process is called as condensation reaction, and it is formed between carbon number 1 and carbon number 4. So when two beta glucose molecules join together for the formation of this glycosidic bond, so you can see in this way, they have to rotate in 180 degree to come to up to the level, and then there will be the removal of this water molecule and there will be the formation. In this way, you can see the structure. In this way, many beta glucose molecules will join together to make one, and so on. There will be the to make one cellulose molecule. So it will be in the form of linear chains. So I have shown only two molecules of beta glucose, but in this way there are many glucose molecules, beta glucose molecules, they will join together for the formation of a single chain. Now, next question is that studies of nitrate uptake and nitrite and metabolism help to provide information to scientists who are investigating ways to increase the yield of crop plants. The first step of nitrate metabolism in leaf cells is the reduction of nitrate to nitrite, catalyzed by the enzyme nitrate reductase. The activity of the enzyme can be studied by detecting the presence of nitrate formed. So we can say that if the nitrate ions are formed, it will show that the enzyme is working well. So researchers have found that adding nitrate to leaf tissue results in an increase in messenger RNA. So when nitrate is added to the leaf tissues, so there will be the increase in the messenger RNA molecules of the gene NR, or the NR stands for nitrate reductase. So state one benefit to leaf cells of an increase in messenger RNA molecules. of gene NR after the addition of nitrate. So in this way, I would like to say that there will be the more increase or you can say increase in enzyme, or we can say that more enzyme will be produced. And which enzyme? This is the nitrate reductase. So if its activity will be increased, so more enzyme will be produced. The next question is, one method used to detect the presence of nitrate formed from the reduction of nitrate in leaf tissues involve using an inhibitor to prevent nitrite from taking part in further reactions in the leaf tissues, immersing the leaf tissues in a solution containing the colorless test reagent. The nitrate from the leaf tissues enter the surrounding solution, changing the color of the solution to magenta. Suggest why using a colorimeter can improve this method to detect the presence of nitrite. Colorimeter, you can see this is the device uh, used in the laboratory. So here you can see that if colorimeter is used in this investigation or in this method, uh, so it will improve the you can say the process, or it can improve this investigation or detection way of detection, because the calorimeter uh, can detect the smallest changes in the color, in the concentration, or anything else. So it can detect the smallest changes. And second, you can say uh, it can also provide the quantitative results. Because it can detect the changes on, you can say, very minute changes. It can observe the very minute changes, or it can detect the very minute changes. So in this way, it can give a better qualitative values, or you can say the accurate qualitative values or quantitative values. So in this way, the colorimeter can be helpful in at this level of investigation. Outline and explain the sequence of events that occurs which allow amino acids to be transported from the apoplast into cytoplasm of a companion cell. And here you can see that, let me go back to the question first. In the leaf, transport of amino acids from mesophyll cells to companion cells involves using a number of different membrane transport protein called amino acid transporter. There is evidence that amino acids can move from the apoplast into the cytoplasm of a companion cell using the same transport mechanism that is used for sucrose transport. Outline explain the sequence of events that occurs which allows amino acids to be transported from the apoplast into the cytoplasm of a companion cell. Students, this is the same method by which the sucrose molecules are transported. So here the same process will be take place over here. That is, you know, that the companion cells and the sieve tube elements work together. The sieve tube elements and the companion cells work together, as the sieve tube elements don't have the nucleus. So companion cells help them to perform their function, because the companion cells have a proper nucleus. So what happens when the amino acids are loaded in the companion cell by active transport? I start writing the amino acids. Acids are loaded by active transport. So in that case, you can see the hydrogen ions, which are also called as the protons, are pumped out of the companion cells into its cell wall. So the hydrogen ions or protons are pumped out of the companion cell into its cell wall by a proton pump protein. And you know, students, the many proteins are present in the cell surface membrane, which act as a channel proteins, carrier proteins, transporter proteins. So they have the different names and they are performing the different functions to transport the different ions and molecules from one side of the cell to the other side through the cell surface membrane. So they are helpful in this regards. So cell wall by protein pump by using ATP, by using ATP as an energy source. And you know, this process, when there is active transport, without energy, the process cannot take place. So the proton pumps are proteins which are found in the cell surface membrane. So these proteins, they create a large excess of hydrogen ions in the apoplast pathway outside the companion cell. So there will be high concentration of hydrogen ion in the apoplast. So they will move back. They will move back into the cell by passive diffusion or facilitated diffusion, you can say. Down their concentration gradient. Down their concentration gradient, so they will be moved back through a again there will be a carrier protein which will act with them. So in this way it carries two substances, because it has to carry the amino acids and hydrogen pumps, so it is called as the co-transporter protein. Next question. And next question is, suggest why amino acids transporters are not needed to move amino acids from the companion cells into the phloem sieve tube element. So here I would like answer of this question is, suggest, question is, suggest Why amino acid transporters are not needed to move amino acids from the companion cells into flowing tubular? Because plasmodesmatas are there, and they can move down their concentration gradient, and by the facilitated diffusion, so you can say. And what are plasmodesmata? They make a connection between the two cells, and due to which the movement will be move down the concentration. By, let's see, or simply we can say the facilitated diffusion takes place. So there is no need for this purpose. Now the next question is, to investigate nitrate uptake, routes can be cut and moved and placed, removed and placed in a buffered solution containing nitrate ions. The root tissues can be analyzed to determine the quantity of nitrate taken up over a set period time, and the part is excised or removed. Roots of the crop plant maize, maize, were placed in three different concentrations of nitrate solution: 0.2, 0.1, and 5.0. So the solutions were maintained at 30 degrees centigrade and aerated to provide a continuous supply of oxygen to the root tissues. Nitrate uptake by the root tissues was determined each hour for 5 hours. So it was observed for 5 hours, and you can see the uh, three graph curves. This lowest one is 0.2, in the center it is 1, and at the uppermost is the 5%, and on the y-axis there is nitrate ions uptake. So it is a fresh mass you have shown. Now question is, show the rate of nitrate uptake is very low initially and then increases for all three concentrations of nitrate solution tested. You can see, first of all, we will observe this, the curve which has 0.2, and you can say it is increases with a constant rate, whereas the other one, you can say, which is why you can say it is first decreasing, increase, decrease. So in this way, this is not a regular increase, you can say. And then we have to compare these two, and this is it shows very lower uptake of nitrates, whereas the 5 shows the highest uptake of nitrate ions. So we are going to write these results in the given space. So we are going to write these, that the 0.2 m mole cubic decimeter shows the constant rate of uptake of nitrate ion as comparison to 0.5 mm cubic decimeter, and the other you can say very slower is 0.2 is showing the slower, whereas the 0.5 shows the fastest uptake rate. So in this way, both shows the different results. Now, E is the nitrate uptake of excised maize roots were investigated under different conditions. Table 4.1 shows details and results for a control experiment and 4 modified experiments, 1, 2, 3, and 4. So these are the 4 modified and 1 control experiment. So temperatures are, you can say the 1 at 30 degrees centigrade, 2 at 3 degrees centigrade, 3, 4, and control, all they were kept as 30 degrees centigrade. So aeration was provided only in 2, 3, 4, and control, not in experiment number 1. So additional substance present in nitrate solution, nitrogen gas bubbled through instead of oxygen in case of experiment number 1, and nitrate uptake was 0.4. In 2, there was no additional substance present in nitrate solution, and the nitrate uptake was 0.6, and protein synthesis inhibitor was present at experiment number 3. So it shows the 1.4, and the antibacterial compounds were present in the fourth experiment. So it shows the rate 10 uptake, or in control experiment it was 10.9. Highest nitrate uptake or fresh mass was by the control experiment. So in this way we are moving towards the question. First of all, the results are shown can be compared by discuss how comparing each of these results with control provides information about how nitrate ions are taken up by the root cells, the factors affecting the uptake of nitrate ions. First of all, how nitrate ions are taken up by the, you can see in the experiments, all the 5 different experiments, 4 different experiments, modified experiments, and one which is control experiment. All they show the different rates of uptake of the nitrate ions. So it means the different uh, substance which were added into them, they also affecting the rate of uptake of nitrate ions. So in this way, I would like to say uh, all these nitrate ions are taken up by the active transport. All they are taken up by the active transport, and for active transport, energy is required. So if the energy is required, sometimes if there is, in any case, if energy is less, so the rate will be affected. So the first will be, in this way we can write that active transport, their uptake is by active transport, and the for active transport energy is required. So if amount of energy is less, so the uptake will be disturbed. So there will be the changes in the uptake of these enzymes. So if we compare any of the experiment with the control experiments, you can say that oxygen in the experiment was not required, or in the other oxygen was required. So in this way, in different cases, when oxygen is required or oxygen is not required, we will move towards the experiment. So in the first experiment, there was no need of oxygen, whereas in all other experiments there was oxygen required. So it also affects the rate of uptake of enzymes. So the different factors, one of them is also one factor was that the ion uptake is by active transport, so it will be affected if energy is not required. The second, we can say, we can make comparison between the requirement of the oxygen. So oxygen, in the first experiment, oxygen was not added, and if it is not added, it means active transport will not take place properly, whereas in control experiment, oxygen is added. So oxygen was added in the control experiment, and if oxygen is added in the control experiment. So what can happen? That is, the process can go well. That is, it shows the highest uptake of 10.9 in control experiment. In control. So oxygen will also play a role in the uptake of these ions and the uptake of the nitrate ions. Okay, now another point which can be written in this way, uh, when we compare all these different results, when we compare all these different results, you can say if uh, the temperature can also affect temperature, temperature can also affect the ion uptake. And you can see in the experiment number 2, the temperature was very low, as it was only 3 degrees centigrade. And whereas in control experiment, it was 30 degrees centigrade. So it can also affect the rate of reaction. And and the other side, we can say that the other thing which can be compared with one another, sometimes, you know, the movement can take place by diffusion. So it will also affect the rate of uptake. You know, it is a slow process. Now we are moving towards the question number 5. And question number 5 is, during interphase of the cell cycle, individual chromosomes cannot be seen within the nucleus. The genetic material is termed chromatin during this stage, which is in the form of fiber network. So it is in the form of fibers, when these fibers make a network inside the nucleus, so they are not visible until this mitosis takes place. So changes occur to chromatin during mitosis so that chromosomes become visible. They are condensed, and due to their condensation, they are visible. Otherwise, they are not visible. In any case, so state what happens to chromatin so that individual chromosomes can be seen during the during mitosis. You can say they are condensed, and when they are condensed, they are visible. Otherwise, they are not visible. They curl up, or you can say they condensed, and when they are condensed, they are visible under the microscope. So you can see the transmission electron micrograph of 2 human chromosomes at metaphase of mitosis. And what is metaphase, students? You know, metaphase is that phase of the mitosis in which the chromosomes, they arrange themselves at the equator in the center of the cell. So when they are at the center of the cells, metaphase, when chromosomes are at the equator or in the center of cell. So which type of they are the actually sister chromosomes or homologous chromosomes. So they arrange themselves in the center in this way. So in this way, they arrange themselves in the center. So, and secondly, two chromosomes shown in figure. So they are in the center, or they have each chromosomes. You can see it is joined in the center is centromere, and these are chromatids. And inside the, and if we see in this way, the outside is made up of protein, and which is called as histone. And this is the histone protein. And inside there is single DNA molecule, DNA, double strand DNA molecule inside. And on the portion which is at the tips of these chromatids is called as telomere. And telomeres is used to protect the genetic information during the cell division and cell replication. So question number 6 is, some people who are infected with HIV have HIV AIDS. State the full term for HIV. This is human. HIV stands for human amino deficiency virus. Following transmission of HIV, only diagnosis of infection and treatment with antiretroviral therapy ART helps to control the spread of the pathogen and prevent HIV/AIDS. Suggest why treating people who have developed HIV/AIDS with ART may help to reduce the number of overall deaths from infectious disease such as the cholera, tuberculosis, and malaria. Suggest why treating people who are they have HIV AIDS, they are treated with ART, ART, which is the antiretroviral therapy. So the people who are treating with these, so they have no infections with cholera, TB, and malaria. So we have to suggest because it is this treatment is about this treatment is about because this ART will reduce the effects of HIV and increase the immunity. And if immunity is increased, will increase, it will reduce the reduce the risk of different diseases, different infectious diseases, we can say that. And due to decrease in infectious diseases, the person can live a better life, you can say, because HIV is not properly working in that case. So the infectious diseases, just like tuberculosis, cholera, and malaria, they will be less effective in that case. Next question. You can see the studies suggest that people who are infected with HIV may be at a high risk of heart disease. One cause of heart disease is the narrowing of the lumen of one or both of the main coronary arteries. A coronary artery bypass, graft bypass, is a surgical operation that uses healthy blood vessels to divert blood around diseased section of coronary arteries. The main choice of blood vessel to use for a bypass graft is known as the internal thoracic artery. So figure 6.1 is a diagram of an external view of the heart to show a double bypass graft. The blood vessels used in the bypass graft are the great symphonious vein from the leg and the internal thoracic artery. So you can see the figure 6.1 in which uh, it has been shown. And now the question is, clearly observe this picture and then draw cross X on figure to show an area of the right coronary artery that has been bypassed. You can see this one. This one, see, this is the area which has been bypassed. So you can put the cross over here. Now the next question. After surgery, the wall of the great symphonious vein becomes thicker. Suggest and explain why it is important for the wall of the vein to become thicker after surgery. So it needs to be thicker because it cannot be brushed in this way, but it can affect the high blood pressure. Its life is increased life of the this blood vessel because it is thicker. It can bear high blood pressure. So it was all about the question number 4, 5, and 6. Please like, share, and subscribe my channel to watch more videos. Thanks for watching.